Hi everyone. Let's continue with our topic types of fermenters. We have already discussed 5 different types of fermenters. Now I am going to talk about the remaining 8 different types of fermenters. Fermenter. What is meant by a fermenter? Fermenter is also called bioreactor. It is used for the fermentation process. Fermenter is a vessel in which sterile nutrient media and microbial culture are mixed and the fermentation is carried out under optimum condition. In this video, I will explain about 8 different types of fermenters, that are tubular fermentor, fluidized bed fermentor, airlift fermentor, packed tower bioreactor, stirred tank fermenter, bubble column fermenter, membrane bioreactor, and finally photobioreactor. First, we can discuss tubular fermenter. It is also called tubular flow reactor. It is a pipe or tube through which the reactants flow and finally converted into products. This fermenter have varying diameter. The tubes may be vertically arranged or horizontally arranged, and the tubes are connected to a pipe system. There are several advantages for the tubular fermenter, that are uniform mixing, tubes are easy to clean, simple construction, and high area to volume ratio. Fluidized bed bioreactor. This type of reactors used to carry out reactions involving fluid suspended biocatalyst, such as immobilized enzymes. In this type of reactor, a fluid is passes through a solid granular material. This is at high speed for the suspension of the solid. The fluid may be gas or liquid. This is known as fluidization. Enzymes are immobilized onto solid granular material. This smaller particle move downward due to the gravity. So it settles at the bottom. The media and oxygen pump through the bottom of the fermenter. So the upward movement and downward movement provide particle mixing and temperature control. Once more, the upward movement of the media and oxygen and the downward movement of the particles provide particle mixing and the temperature control. Okay. Next, we can move on to stirred tank fermenter. Stirred tank fermenter is also called continuous stirred tank reactor, or simply CSTR. C for continuous, S for stirred, T for tank, and R for reactor. It consists of a cylindrical vessel with motor driven shaft that supports one or more agitators. or impellers. Shaft is fitted at the bottom of the bioreactor. Impellers provide proper mixing of the microorganism and media. Baffles are attached to the wall of the fermenter. Air is added to the culture medium under pressure through a device called sparger. The sparger may be a ring with many holes or a tube with single orifice. Okay. So spargers are mainly two types: ring sparger and tube sparger. Sparger and impellers provide gas distribution throughout the vessel. Air lift bioreactor. This type of bioreactors are mainly used for aerobic bioprocessing technology, and also it is used in wastewater treatment, SVP production, methanol production, etc. In airlift fermenters, the medium of the vessel is divided into two interconnected zones. This division is by means of a baffle. One zone is called riser zone and other one is called downcomer. The air is pumped through riser. It allows the upward movement of the media with gas. The media downflow occurs in downcomer. So the upward movement of the air and downward movement of the media will result in dispersion. Next one, membrane bioreactor. This is mainly used for wastewater treatment procedures. And it is a new approach to the fermentation technology. One example for this is the ethanol production. Membrane bioreactors are hybrid systems comprising a bioreactor for bioconversion and a membrane device for the separation. Okay. Note the name membrane bioreactor. So the membrane device is for the separation and bioreactor is for the bioconversion, that is the product formation. Then we can discuss bubble column bioreactor. The air is introduced through the bottom of the fermenter. The airflow provides oxygen transfer and mixing of the contents. Bubble column bioreactor is a cylindrical vessel. The air is introduced through the pipes or plates. Okay. So the air is introduced through the bottom of the fermenter. Next one, packed bed bioreactor. In this, cells or enzymes are immobilized on larger particles. So it cannot move through the liquid. So it is settled or packed in the fermenter. Hence the name packed bed bioreactor. The solids used may be porous or non-porous gels. A nutrient broth flows continuously over the immobilized biocatalyst. The products in the packed bed bioreactor released into the fluid and removed. Last one is Photobioreactor. These are specialized bioreactors. It carried out by exposing to sunlight or artificial illumination. Artificial illumination require high cost. Okay. They are made up of glass or transparent plastic. An array of tubes or panels constitute solar receivers. Photobioreactor operate at a temperature of 25 to 40 degrees Celsius. The culture can be circulated through solar receivers by centrifugal pump or airlift pump. Okay. This is about types of fermenters or types of bioreactors. We have discussed about 13 different types of fermenters in 3 different videos. Okay. I hope you understood this class. Thank you. Thank you all.